Okay, so it is 9 o'clock. I'd like to call the meeting to order. Looks like we have a quorum, Mark.
I can take roll if you'd like me to, Scott.
Yeah, we have 8 voting members.
Okay, we should be good shape then.
Mhm.
We don't have any minutes, I believe, from the last meeting, do we? This has been a special meeting. We haven't have the meetings, the minutes yet from the last meeting.
That's correct, Mr. Chair. We'll have those minutes prepared and presented at our next regular meeting, if that
That's good. Okay.
pleases the board.
No minutes for the last meeting to cover. So just say we've got a lot to cover here this morning, so we need to stick to the agenda best we can and try to get through on time. As we review these 7 proposals, keep in mind that there's, as I roughly calculated, there's about $748,000 in requests, and we've got about 144, something like that, available. So we can fund some combination or whatever, or we don't have to fund any of them. There might be other projects surface later we want to do. So we've got a lot of flexibility, I guess. And keep in mind that these projects, for the projects that we're going to be reviewing today, the funding is from House Bill 3012, which is fisherman money, agreed to fund this research for the purpose of restoring, increasing harvestable fish returns as soon as possible, while simultaneously protecting the wild fish. So all these projects have merit in their own way, but we can't fund all of them. And so kind of keep prioritizing in mind when we go through them, and kind of keep an idea of which ones you would like to fund, and hopefully we can get the agreement at the end of how to do that. But we will need a motion at the end, motion to fund one or 2 or parts of them, and how much money that we're going to allocate to them. So kind of keep your notes and keep that in mind, because when we get to the discussion period in the end, we're going to have to figure that out and make a motion before the end of the day, and limited amount of time. So we need to keep clicking to get it done. So if you kind of keep that in mind and keep your notes along the way. If we have just a minute, Selena, could you give us a quick update on where we are with the director? I saw a note that, go ahead.
Thanks, Scott. Yeah, I have bad news, everybody, which is that Dr. Narum has backed out. He got a counter offer from Kritvik, and he's decided not to come. This is pretty frustrating, I think, because we've been doing a lot of work to get things set up and come to an agreement on the things that he requested for his startup and so on, and a job for his wife and a whole bunch of other stuff. And it's all fallen through now. So we have 2 candidates who are still in the pool from the original search, and we are in contact. One of them, we've already started the process of setting up the interview for Dr. Mark Christie, who is at Purdue University. And then the other candidate we'll be talking to is Seth White, who is also from Kritvik. Both of these are former students from OSU. Mark was in the zoology department with Mark Hickson, and Seth was one of Hiram Lee's students. So you might remember Hiram. So I'll be working with ODF&W and letting all of you know what the schedule is for that interview, those interviews. And we'll keep our fingers crossed that that goes well, and that we are able to identify a director. So sorry about that. And I'm really, yeah, like I said, pretty frustrated about it, but appreciate the time and energy that many of you put in to attend or watch the seminars and things like that. So we're hoping that you're able to join us and help us with that process once again.
Thank you. And would you please keep us informed, or if you have any resumes or information, send it out to us?
We'll send those right away. Yes, we just need to confirm that both of those candidates are coming, and then we will send out that information. Not coming, I should say. I'm sorry, Scott. The interviews will be by Zoom, just as the first two interviews were. So when I say coming, I don't really mean physically.
I went to school at Purdue, but I never caught a salmon back there. I guess I got them up in the lakes now, though. Okay, with that, I think we're ready to move on. Next on the schedule, Mark, keep me informed if I'm slipping off here, but at about 9:10 now, we've got Dr. Couch presentation, so we might as well move on with them.
That would be me. Okay, so bear with me for just a second. I always struggle setting up screen share via Zoom. So
It's looking good. Yeah, good.
and then I just wanted
Looks good.
to presenter view. So can you see my slides, but not my notes, I'm hoping?
Correct.
Yes.
Okay. If you can see my notes, then there's no point in me talking. So if this looks good, I'll go ahead and get started. So my name is Claire Couch. I'm a postdoc at Oregon State University. And my PI, as well as my co-PI on this project, is James Peterson. So just because I'm kind of new to this group, I wanted to introduce myself really quickly. My background is primarily in disease ecology and bioinformatics. And as many of you know, Jim is the unit leader of the Oregon Cooperative Fish and Wildlife Research Unit, has decades of experience developing decision support tools for fish and wildlife conservation and management. So he's really helping me translate some of my skills into applications. So that's just a little bit about us. And now I'm gonna talk about a proposal, which is titled Optimizing Rearing Conditions for Hatchery Production and Conservation of Pacific Salmon. First of all, I wanted to frame this proposal in terms of the OHRC mission, as well as the original RFP that we wrote this proposal in response to. So as you all know, part of the OHRC mission is to understand mechanisms that may create differences between hatchery and wild fish, and to develop approaches to manage hatchery fish that conserve and protect native fish. Then in the RFP, one of the priorities was to identify traits that are under selection in salmonid hatcheries and identify ways that fish culture techniques could be modified to ameliorate selection. So the approach that we are proposing here for addressing these points is to understand how hatchery rearing conditions exert selection on the gut microbiome of Pacific salmon, and to use this information to develop culture techniques to improve hatchery fish health and ultimately reduce the impacts of hatcheries on wild fish. So I want to briefly provide some background on gut microbiome research in general. So essentially, the gut microbiome is a huge collection of microorganisms that reside in the gastrointestinal tracts of all vertebrates. We actually knew fairly little about gut microbiomes until the early 2000s, when some new sequencing technologies and bioinformatic tools allowed us to start studying the gut microbiome in great detail and at very low cost. And the gut microbiome has become hugely important in biomedical science. There are entire journals dedicated to this subject now, and Oregon State University is actually kind of a hub of microbiome science. So I am lucky to be part of that community. So the gut microbiome has been found to play really key roles in host immunity, nutrition, even things like mental health and behavior, and just numerous other functions. It's also fairly easy to measure and manipulate the gut microbiome. So it's currently the target of new diagnostic approaches and personalized medicine in the fields of human health. And then importantly, many of these same functions that have been discovered in the human microbiome also hold true across other animal species. So as I will get to in a second, a lot of what we've learned in humans can be applied to other animals, including fish. Yeah, in the past couple of years, the gut microbiome has become a major topic of international interest for fisheries and aquaculture. This graph shows how the number of studies on fish gut microbiomes has increased in the past 10 years. And the blue bars here represent aquaculture species. Just to illustrate this, I recently attended a large international conference that was dedicated solely to fish gut microbiome research, and was able to connect with many scientists from all over the world who are exploring ways to use the gut microbiome to improve aquaculture. I'm not going to summarize all of the hundreds of studies that have now been conducted on the gut microbiomes of fishes, but I am going to just highlight a few key points. Causes of variation in the fish gut microbiome include diet, environmental conditions, antibiotics, and host genetics. And altering or manipulating the microbiome has been shown to result in changes to immunity and disease susceptibility, increased or decreased growth, behavior, and many other important outcomes that I'm not going to go into. But I would invite you to explore that literature if you're interested. So the microbiome is emerging as an essential aspect of fish health and one that can be easily monitored and manipulated. I also quickly want to highlight some of our previous work, which is not yet published, but I'm actually about to submit it for publication. So we conducted an experiment in which we simulated chronic stress in juvenile Chinook salmon, when we found that this majorly affects the gut microbiome. In fact, we found that gut microbiome variation can actually predict mortality in fish that are stressed. We found similar patterns in adults and juveniles, suggesting that the gut microbiome is a powerful tool for monitoring chronic stress and mortality risk in Pacific salmon. And as a next step, we want to explore different ways to modify the gut microbiome in a hatchery environment to create fish that are healthier and more resilient. Okay, I guess I had that slide twice. So the overall goals of our proposed work are to understand the differences in the gut microbiome between hatchery and wild Pacific salmon, and optimize rearing strategies to enhance the microbiome for fish health and survival. Finally, we want to build on our previous work that I just described to develop low-cost, non-lethal monitoring tools for fish health. So here are the specific objectives that we included in our original proposal. First, we wanted to evaluate the effects of different diets, probiotic and antibiotic treatments, and the physical rearing environment to produce healthier, more resilient, and potentially more wild-like fish by affecting the gut microbiome. And then we also wanted to build on our previous work and develop low-cost microbiome-based monitoring tools for immunity, growth potential, and survival. So to achieve these objectives, we would need funding to sequence microbiome samples collected during a diet manipulation experiment that we already conducted, as well as conducting some additional experiments with probiotics, antibiotics, and different environmental conditions. And finally, we propose to conduct experimental releases to assess post-release survival. So to do all of this, we originally requested nearly $140,000 from OHRC, in addition to some funding that some pilot funding that we have from other organizations. Given the changes in the budget situation since the original RFP, I also want to present an alternative that would narrow down the scope of this project quite substantially, but would also reduce its cost. So in 2021, we conducted a diet manipulation experiment in which we fed juvenile Chinook salmon either a standard bio-Oregon hatchery diet or a diet that is designed to mimic what wild fish would be eating. And then we collected samples from the gut microbiome over time and measured outcomes in terms of immunity and growth. So we would be able to use this data to meet a couple of the objectives that we originally outlined. We received a small pilot grant to sequence and process a small number of the samples that we collected from this experiment. But with the addition of $50,000 from the OHRC, we could sequence and analyze this entire data set from the experiment we already conducted, which would make us much more likely to generate useful information for hatcheries. So because this experiment has already been conducted, and because our team has extensive experience analyzing this type of data, we would expect useful results within a year of funding. And then we would hope to eventually obtain additional funding to address some of the other objectives from our original proposal. So really quickly, I just wanted to show you a schematic of the general framework we used for the diet study, and which we would also use for additional experiments. So first we would rear juvenile Chinook salmon under different conditions, collect microbiome samples and body measurements over time. And then at the end of the study, assess growth, changes in immune gene expression, and microbiome similarity to wild fish. So we can then link these health and performance outcomes with the gut microbiome variation. So ultimately, we would want to conduct experimental releases to compare survival and post-release performance of fish with different microbiome communities. Okay, so just a couple of the expected results and benefits of this work. We would anticipate generating important information for rearing healthy hatchery fish and reducing operating costs and minimizing the impacts of hatchery fish on wild fish. So by optimizing the gut microbiome, we can improve hatchery practices to reduce disease outbreaks and disease transmission to wild fish. We could also find ways to improve growth, which could improve post-release survival. And then these methods could contribute to improved smolt adult return rates, thus reducing the number of fish that would need to be released. And that could lead to benefits, including reduced hatchery operation costs and reduced pressure on wild fish. And then, as I mentioned before, this project would also contribute to developing non-lethal microbiome-based monitoring tools to predict fish health issues before they become major problems. This tool could be adapted to hatcheries across the Northwest, as well as potentially being adapted as a tool for non-lethal monitoring of wild fish. And then finally, I just wanted to point out that this work would be complementary to some of the studies that OHRC has funded in the past, in particular, some of the genetic work. As I mentioned before, host genetics shape the gut microbiome, and in turn, the microbiome can regulate how host genes are expressed. So essentially, the microbiome can turn host genetics up or down. So I would greatly look forward to connecting with and collaborating with some of the outstanding researchers that already work with OHRC. And I would expect that we could develop some really productive collaborations. All right. And so just to finish, I put a couple of key points up here and I would welcome any questions from you all. I might hand some of them off to Jim because he's really more of the fish expert than I am, but if I can answer all of your nerdy bioinformatics questions.
Any questions from board members for Dr. Gooch?
Not this time.
Well, if there's no further questions, we can move on. Are there any more questions for Dr. Couch? Okay, that takes us on to Dr. Matt Falsey models. Thanks, Claire.
All right, thank you, everyone. Can you see my screen all right? Picture of a fish.
Yep, we got it.
Yes,
Thank you.
yes.
Okay. Well, let's see here. Sorry, I'll just go from this mode here. So my message essentially is that I believe that applied science is rarely ever applied. A gap between publication and public action is huge because the features of actual decisions are usually ignored by scientists. And we're still, I don't even see how we could possibly determine what needs to be known without understanding how knowledge and uncertainty get wrapped into decisions. Indeed, I posit that prioritization of information needs without an explicit model of how that information will be used often leads to wasteful spending on research and information. So I'm here to pitch a hatchery management tool. And this is probably outside the box of your expectations because there's no empirical science here. There's no new information that I propose to generate about nature itself. Rather, the focus of this work is to clarify the management context and the management decisions. And I am positing that this sort of information is needed not only to prioritize future information needs through the OHRC, but also to better understand how existing science is being used to make actual real-world hatchery management decisions. So hatchery management is an exercise in risk management. And we want to find the sweet spot that maximizes fishing opportunity and minimizes risk to natural origin fish. But unfortunately, you can't have your cake and eat it too. There's a fundamental trade-off that's inherent here. It's unavoidable. So how is this trade-off actually implemented in actual practice? Right? How do we balance the fishing opportunities with risk to natural origin fish? There are tools, scientific tools that are available, existing information and tools that can be used to inform some of the risks and some of the rewards. But there are also baked-in assumptions that are going to be inherent in this trade-off that people will often implement with completely internal mechanisms, gut feeling, if you will. But I am here to propose a hatchery management model because with such a thing, we can become totally transparent in how we make decisions about whether or not hatcheries are meeting our needs, whether they are correctly balancing the risks and rewards. And we can also use this model then, as I've indicated, to determine what are the critical information gaps and how that relates to our ability to attain our management goals. So the risks to domestication selection have been analyzed and studied with formal models. The work in quantitative genetics done by Michael Ford quite a long time ago is kind of captured here, where there's hatchery and wild fish that are exchanging individuals across these two structured populations. There's a bunch of parameters and gobbledygook in there. And what pops out of it essentially is an estimate of fitness of the natural origin fish. And this is really important for making management decisions because this informs how much risk our hatchery fish are imposing on natural origin fish. There are a number of parameters here. There are a number of assumptions in here. And it is becoming clear about what these parameters are, what their values are, and what their uncertainty ranges are that informs our understanding of risk to the natural origin fish. It can be done not only with closed form math that Ford developed, but also through simulations. In an allele-based model that I created, it's possible to relax many of the assumptions that are sort of needed for the mathematics that Michael Ford developed. And when these assumptions get relaxed, slightly different results begin to appear. So with the figures down here is just some runs from these simulations where there's an initial population of natural origin fish in the blue line, and their phenotype is being plotted along the y-axis. And at a certain point in time, a hatchery is initiated, and they're plotted there in green. They have a different optimum phenotype, right? There's an inherent difference in the environments between natural and hatchery origin fish, which is unavoidable. And so through time, the hatchery origin fish begin to evolve closer to their optimum that is found within the hatcheries. And then at a certain future time, maybe there's some climate change that goes on that causes the natural origin fish to have a different optimum phenotype. So that you can kind of see the bend in the red line there. And in the case on the left, the natural origin fish adapt and evolve, and they keep up with this rate of change. So this is a scenario of climate change where the natural origin fish are able to maintain their adaptive potential and keep pace with changing environments. But this is under a scenario where PHOS, proportion of hatchery origin spawners, is 0. On the right-hand figure, there's a different scenario where proportion of hatchery origin spawners is increased to 30%. And in this scenario, the model indicates that the natural origin fish cannot keep pace with climate change. And because this is an individual and allele-based model, we also get demographics out of this. And because of fitness declines due to the exchange of genetic material in two different environments, the demographics fail. So there's ultimately a extirpation of the population in this scenario. So both via allele-based modeling and the closed form mathematics from Ford that I provided or showed you earlier are tools for understanding the magnitude of risk, right? But this is not the whole picture, right? There's also, we acknowledge that there's some risk. I posit that we need to be clear about how much in order to really make good decisions, but there's also a benefit to having these hatcheries as well, right? And that's through fishing opportunity. So what I'm proposing is a management model kind of embodied with this example here that blends together the risks and the rewards. And if we study this thing really closely, and this is just an example, but the output of this on the right is just this hatchery program success. Is it good or bad? And this is, in this particular case, the ultimate measure of whether or not we're attaining our goals, right? And so it's a probability of a goal attainment, if you will. But there are two main things coming into that final node on the right-hand side. It's the wild risk, and everything that kind of flows into that box is related to the stuff I was just showing you before. But the other box flowing into the final node there is the fishery quality. So this is the reward part of having a hatchery. And there are many elements of fishery quality that include not only the opportunity to go fishing, but the actual catch of fish too. And there are further things like the size of the fish and the durations of the seasons, and a number of elements that ultimately inform fishery quality from the perspective of anglers. I just have two examples here of things that could inform that. Ultimately, it's a question of social science, of how people respond to and enjoy these fisheries. And what I'm proposing with a model like this is an explicit method of blending the risks and the rewards of a hatchery. One thing to point out in here is that if you follow the wild risk side backwards a little bit through the arrows, you get to a point where there's this node in there, this box about the wild fitness, and under strong selection and weak selection. These are some of the parameters that were built into the Ford model that I showed you, and it's also baked into the allele-based modeling that I have done. And if we toggle between different assumptions in the strength of selection, as I'm going to do right now, you can see how this propagates through this network. And we basically have a hatchery that goes from being 70% good to 30% good. meaning that we have radically different understandings of how this hatchery is performing relative to our desires of the balance between risks and rewards based on one simple assumption about the strength of selection. This to me indicates that we really have no way of evaluating the success of our hatchery until we're being clear about what we believe the strength of selection is. And in this case, the strength of selection on what is something you might obviously be interested in. And it's not just one thing, it's a whole bunch of things, right? So in this case, we're just looking at a univariate single phenotype, but we can generalize this to multiple phenotypes. The key thing here, in this particular case, I'm merely isolating one thing here, the strength of selection. And I would point out that Dr. Bluen is already studying this. And so I've written in my proposal the desire to work with him to help parameterize this model so that we can, instead of just toggling crudely between strong and weak, we can actually come up with a range of uncertainties in the strength of selection that's been informed by his science and bake it into a model like this. So in conclusion, the model that I'm proposing here is not new empirical science. It is, in my view, a necessary management tool that is really going to be something that ODFW has to have buy-in on. If they are not willing to make actual hatchery management decisions with a tool that we develop, then there's no point in doing this project, right? I'll tell you that right now. But if we can get managers from ODFW to get buy-in on being explicit in the way in which they make these decisions, then I propose to you a method in which we can use existing hatchery science. And once we've specified all of our assumptions in a model like this, we can use it to probe what are the new science and information needs to close critical information gaps. So I'm way over budget on this, and that's a conversation we can have. There are things I can cut out, and indeed even what I've just provided you here cuts out a lot of things that were in the original budget. There's some unfortunate overhead associated with my affiliation with University of Idaho, but if this is something that sparks interest, I'd be happy to circle back around later on revisions to the budget that I've given you. I'll end there. Thank you for your time.
Thank you, Matt. Do we have questions from board members for Dr. Falsey?
Yeah, Matt, this is Steve Jacobs. I had one question. So is this model primarily to identify information gaps or management direction in terms of strategies?
Yeah, I think it can do both in the sense that it can tell you, in this particular case, the one I've just showed you, what are the PHOS and PNOB, optimal PHOS and PNOB to get this hatchery at, to maximize its being quote unquote good. Obviously, as you increase PHOS, you're going to also increase fishing opportunity, right? Like as you make more production, more hatchery production, you get more fishing opportunity, but then you also get an increased PHOS and hence risk to the natural origin fish. So in this sense, once we're clear about how the risks and rewards are being blended, we can use it to optimize hatchery production, optimum in the sense of that balance between risk and reward. So in that sense, it's a hatchery management tool for actual real world use on how to optimally run a hatchery to attain our stated objectives. But it also does the other thing you mentioned, Steve, of identifying information gaps, because there are assumptions in here. Science informs parts of it, but then assumptions are inherent in a complex decision like this as well. Science isn't everywhere. So one thing we can do, which I haven't really described yet, but you've keyed into, is that we can do sensitivity analysis on a model like this to determine which of the assumptions that are in here have the greatest impact on our decisions about hatchery management. Again, here it's just on PHOS and PNOB, but there are other levers within a hatchery that are available. to be turned or pulled, if you will. And these are things that my team and I are proposing to develop with managers at ODFW to get their buy-in on the use of a tool like this. And there's need to be comprehensive about all of the management levers. And I've just kind of focused on 2 of them here. So in that sense, Steve, yeah, I think it's both a tool for actual use, but then it's a tool that can be manipulated and analyzed to determine what are the key uncertainties in hatchery management that should be filled with science.
Okay, thanks.
If there's no other question, we'll move on.
Well, I had one last one, Scott. This is Seth Eisenberg. What would be the time frame you'd be able to make a revision, or if that's feasible relative to the budget kind of allocation that we have on the table and the delta with your current budget projection?
Yeah, so I think there are 2 questions in there. I can get you a new budget within a week. And then that new budget will basically reduce the time frame of the whole project as well. That's how I'm going to get the cost savings. And I'm imagining basically chopping this thing in half. So the deliverables coming out in roughly a year. And that should basically have the cost. But I can get you details on that within a week if there's interest in this after you've had your deliberations.
Thank you. That's important context. Appreciate it.
So as a brief follow-up, Matt, then is it fair for the board to, you might be able to bring this to about 100K?
Yeah, that's exactly right there. And it will, the new proposal, this was only hit me like a week ago that I was basically twice the budget. Again, I have like an overhead of 50% here. So that's part of the problem. But this is news to me of only about a week ago. So you will receive, if you're interested, a new proposal that will tell you exactly what's going to get cut out and what will remain. But the things I've provided in this presentation here are all things that can remain within the new proposal. That would, in fact, meet your budget. There's no point in doing it if you don't have the money. So I'll make sure to get you a product that hits the budget and detail in the proposal exactly what that is.
Thank you.
Okay, thank you. Michael Banks, you're on.
Oh, good morning, everybody. Thank you very much for this opportunity to present to you. And before I actually open this PowerPoint, I just want to show you the current technology that's used for monitoring hatchery salmon in terms of their abundance and returns and their behavior in the ocean. So this is a coded wire tag reader behind me. It's one that I borrowed from Sandy Hatchery. Fish are passed through this opening over here, and by gravity flow through. There's a metal detector in here, and if it has a coded wire tag, then one of the doors will open and the fish is passed to the bin in which coded wire tag fish are collected, and if not, it goes to the other bin which is headed for food share. So let me open what I have to present as a complement to that technology. Oh, that's interesting. Wasn't planning on showing you a story about elephants. You're seeing some salmon now?
We were seeing salmon before.
Oh. This is terrible. Let me try again.
Michael, this is Carl. I learned last time. I think if you go to slideshow rather than the little icon at the bottom, go to the top, slideshow, and then from slideshow go to start at the beginning, and that should work. At the top, you should.
Sorry, guys, I'm struggling with tech here a bit.
At the very top, you should have a link to slideshow.
How about that? Are you seeing salmon?
Yes.
And now are you seeing presentation mode, or what are you seeing?
Yeah.
We're seeing your presentation. It appears that you've got, it's capturing our, there we go.
All right. So my title is Sampling Thousands of Salmon to Facilitate Hatchery Management. And just in terms of general concept for my mission, you guys know me pretty well, but perhaps some not. Our lab studies the genetics of life history diversity in salmon and other fishes because they are a vital component of the splendor of life in our region. Hatchery managers have a pivotal role in preserving that heritage. And our innovation is to help hatcheries and communities better protect that gem. So what is the problem? Hatcheries support a vitality of salmon where fisheries are among 7 different species embrace tremendous diversity. This needs to be evaluated, protected, and maintained to guarantee futures of fish and fisheries. Yet the differences are sometimes hard to discern just by eye, especially when we have different mating strategies or different life history patterns within the same species. So currently, tagging using the technology that I have behind me right now, such as fin clipping or metal and other tags are used, but they have shortfalls. And just to overview some of those, they're costly and they take time. Very often, fishes are managed on last year's data or maybe even distribution data that may date back to the 80s. The ocean is different today in terms of how it was in the 80s. Also, because it's costly, we can only sample about 10 or 20% of the total run. So there's large estimation areas, especially given the small recovery we get from a salmon life history. There's also other side effects like straying or fin regeneration, less fist mating, offspring survival, etc. Some of those things very familiar to you. So we, thinking about that, have a solution and something I'm calling a fast sorter and tagger. As I mentioned, we'd like to complement this Northwest Marine Technology tunnel reader, like I have in the background here. It'll be a tool for hatcheries in state and federal hatcheries, and also has application in markets because market fraud is a big issue in terms of fisheries. And in fact, I learned from some of the salmon commissions that it's an issue for them in terms of access to markets. For example, Europe will only entertain markets of fisheries that have guaranteed certification on their stocks. So basically, this tool will sample DNA and enable precise ID for all the fish as estimates using a genetic technique we call parent-based tagging to better guarantee sustainability in all aspects of the life history of salmon and overcome the tedious sampling for DNA by hand, expansion uncertainty, and other side effects of the coded wire tag or other fin clipping technologies currently used. So this is a sort of a very rough concept design of what we would like to do. This blue box here is like what you see over there, and I'm surrounding with some of our ideas. We would like to have a digital camera positioned above and observing down so that it can image and locate the tail of the fish, which may be in different positions. The punch and a tray beneath the punch would sample and collect the DNA. And through software integration and a motherboard, we'd be able to make this all happen pretty efficiently and be able to record besides gathering the DNA, also color, weight, length, and other details about the sample, and in association with the coded wire tag reading. We'd obviously position this channel in some form of channel which could move up and down. For example, in Coho, this last year, the jacks were from about 28 centimeters to 34 centimeters, and the adults went all the way up to 70 centimeters. So we obviously need to be able to efficiently move up and down according to the size of the fish. And then, of course, the doors would be just the same as what I have behind me. If it was a coded wire tag fish, it would go into one bin. If it was a regular fish with no tag, it would go into the food share bin. So who are we, and what, I guess, the project and team has expanded a little since what I proposed to you. I think it's about 6 months ago when that proposal was written. You know me pretty well. I've been in this field for about 27 years and what I do. I've been working with Ryan Queen from Sandy Hatchery now for about 4 years doing the Mate Choice project. He's part of this too. I'm working with Drummond Biles, who is an engineer managing the iLab here, about 50 meters away from where I'm sitting right now. Sean Niram, who was the director in consideration, has decided to remain at Columbia River Intertribal Fishery Commission, and he will be very useful to us there, still in that position. He's part of the project. Two NOAA scientists, Carlos is down in Santa Cruz, and Mike Ford is up in Seattle. And then a U.S. Fish and Wildlife Service geneticist, Christian Smith, who's manager at the Abernathy Fish Technology. We hope to recruit 2 or 3 undergrads and 1 graduate student. Our current plans and needs, obviously to work with the iLab. This is a multi-million-dollar investment and actually has other funding to help co-support students. One of the students supported by the Roundhouse Foundation has indicated interest in this project, and we're beginning to work with him. We'd also like to bring another, I mean, obviously the camera and the plunger and all these sorts of things will need lots of effort. I'd also like to work with somebody from the business school to ground truth and work further on the business model I'm developing, and a graduate student to pull this all together. Explore partnerships with Northwest Marine Technology. My NOAA colleagues say they have approached them in the past and they weren't interested, but maybe they would be more interested to work with us in an academic affiliation. I'd also like to motivate and work further with state and federal hatcheries, regulatory management folks, and certification and marketing potentials. I actually estimate that it'll cost a lot more than the budget you're considering to pull this all off completely. So I already have another grant into OSU Accelerate. They're going to be deciding on that soon. And I'm considering NSF SBIR. I think this project will probably need about $500,000 to pull off completely. I'm working with OSU Legal Patenting Office in terms of patenting some of these ideas. And of course, I'd like to look at potential international markets, especially with regard to the access to European markets and Asian markets, as I mentioned earlier. So what would be a difference as an outcome of FST? They would be tighter, 100% fish of the tag would be tagged, estimates, which would be helpful in terms of hatchery in the ocean and global markets. There's a cost advantage to this. You just get the DNA from 2 parents and you have tags on all their offspring. That's a wonderful sweetness of this technique. Obviously, if you don't put coded wire tags in the heads of all the juveniles, you may overcome some of the side effects of that technique as well. And for us right here, you know, we've invested a lot in 1 million fish that are now in rearing and out of the ocean because of our project, and we'll have much better numbers for them if we could sample them efficiently at Sandy next year, or actually in November this year, November the next year, and November the following year. And this could be applied to quick and complete ID and other experiments at hatcheries, rivers, and the ocean. And basically just present another tool to help hatcheries preserve the vital component of the splendor in our salmon in the Pacific Northwest. And that's what I have to share with you on our ideas. Oh, I guess budget came up. The budget that I proposed to you began in January and had funds assigned to these first 3 months that have of course passed. So I don't know exactly, but I would say that's probably about 30,000 that could be saved from that, which would bring our budget down to about 100,000. As I mentioned, I think I need a lot more to pull this whole project off, and I am actively pursuing other funding mechanisms as well. Any questions?
Okay, thanks, Michael. If there's no further questions, we'll move on to Dr. Peterson, Integrated Values.
I did have a quick one. I'm sorry, I couldn't get off mute fast enough. Scott, can you give me 1 second? Michael, on your commentary related to your overall budget versus where this particular piece, if we were to fund at the level you'd requested, kind of takes the ball down the line. Can you articulate that a little bit, just to make sure we kind of understand the, sort of the modular aspect of it a little better?
Yeah, thank you for that question. As I indicated in the proposal, and I think even, in this time frame, I really would like to get a prototype to test in November this year, and then have a tested and much more rigorous product to use in November the following year. And of course, we would use it the following year as well. But we could certainly come up with a prototype. We already have that undergraduate interested in working on the project. And I'm thinking of bringing in a second one over the summer, because I really need the sampler in November this year. And so I'm very motivated.
Right. Thank you. I had noted that in the proposal, but I wanted to make sure that that it was clear kind of where where this fits into the bigger frame. So I appreciate the clarification.
To be clear about the bigger frame, that would be to create this product and put it out there and actually support it. And there's lots of complexity.
Hence the SBIR context, right? Where that then's become a for-profit entity on that side.
Yeah, and we are still, I'm still considering and getting advice about maybe working with Northwest Marine Technologies. That would actually make it a lot easier if they welcomed us. They're already in the field and have this, well taken care of.
Yeah, Michael, I had a quick question. Steve Jacobs. It's been a while since I've worked with Codewire tags, but this sampler would only be for dead fish, or would it work for live fish as well?
I hadn't thought about that. I guess you could push live fish through there, and they, by hand we can do that. We have done that. We live clip juveniles and let them go. And in the winter run project I was involved with in California, we actually proved that had no effect on their survival. So yeah, no, one could do that.
Well, it would be more applicable, for like fish traps or barriers or whatever.
Yeah, thank you. That's a great idea. Obviously, that would be another reason why I would need to have very efficient integration with sliding in position and taking a snip and opening the door in time. We can't keep the fish out of water for that long. So that's great.
Okay, thanks.
Are we ready to proceed?
Yes, go ahead.
Great, thank you. So I'm Jim Peterson. I'm the Oregon Cooperative Fish and Wildlife Research Unit. So that means ODF&W is my cooperators, OSU are my cooperators. Of course, I am a USGS scientist. So that kind of brings in everything that the OHRC is all about, this sort of collaboration. And I'm going to start at the beginning of the story. I'm going to talk about decision making like Matt talked about, but Matt kind of starts in the middle of the story there. And that's a really dangerous place to start, because what you find out is people talk past each other. They're not talking about the same thing when you start in the middle of the story. I want to start at the beginning of the story so that we have a common understanding and shared objectives. So that's really what our project is basically about. So the motivation for our proposal was basically the results of the poll that was taken during that OHRC workshop, where we had a bunch of scientists come in and everything else. There's this one right here: do strategies manage difference between hatchery and wild fish optimally satisfy objectives, and then population viability analysis and other modeling to understand alternative outcomes. 20 votes to 16 votes. So a model. Talking about a model, but it needs to be useful for decision making. Proposal involves implementing structured decision making. It's just a process. It's a process by which we first find our shared objectives, right? What do we care about, right? It helps us focus on those things. We break it down into our objectives, the potential actions that we can take to fulfill our objectives, and then we build a model that links those two things together. And we can use that model to do things like prioritize management efforts, but also prioritize research, prioritize monitoring. And I hear this a lot. Don't we already this already? We kind of break down and think about our objectives, and we just implement these actions. I'd say no, we don't. There's a lot of confusion over objectives, right? There's means objectives, which are the things that you think you need to achieve in order to achieve a much greater objective. And I'll give you an illustration of what that is. And many times people get tunnel vision, right? And when I hear you folks and I've interacted with you in the past, you're kind of talking past each other many times because you don't have this shared understanding, these shared objectives, because you haven't gone through the process, right? So there's also confusion between goals and objectives and uncertainty and technical uncertainty and how does the system work and things like that. And then there's also often a failure to incorporate research and monitoring formally into decision making, right? How do you use it specifically to make a decision? That's really inefficient use of resources. So let me give you an illustration. Let's say I have this objective of increasing salmon harvest or fishing opportunities or whatever that happens to be, right? Well, one way of getting to that objective, so a means to that end, might be increasing hatchery production. Okay. For another means to that objective might be increasing natural production, right? Because what you really care about is increasing salmon harvest, right? Or salmon harvest opportunities or salmon population size, whatever it happens to be. And how do we increase natural production? Well, maybe one way of doing it is build out this whole thing. And that's part of the process. So then we begin to understand how other people are thinking, how things work, right? And we're figuring out what their objectives are and what are our shared objectives, and then how can we achieve those things. So why do we model decisions? Why do we use a model? Well, we're always using a model. It's in our ears, right? That black box, right? Model decisions, we want really a transparent means by which conveying information about how we think the system works, right? So they're quantitative methods, but they don't take over, any sort of intuitive, subjective things of decision makers, right? They always have the ultimate say, but they do facilitate communication. I like to think about it as, you think about movies and TV shows. You have the MacGuffin. MacGuffin's the thing that the story revolves around. That's the model. The model focuses the discussion, right? You're talking about this thing and how you think the system works. And sure, it's a mathematical thing, and I'm not going to show you math. You don't need to see math right now, all right? Just talk about how we think things work and put together a bubble diagram. It's great. So the alternative to that is let's keep doing what we're doing, right? Let's keep talking past each other. Let's keep having angst and conflict, right? So this black box, right? We have two people who have perhaps the same objective, right? They want to maximize fish harvesting opportunities. And maybe they have two different mental models of what that means. So hatchery fish released, maybe they think it's good at some point and then it becomes bad at another point. Whereas this person thinks, no, the more hatchery fish you release, the more fishing opportunities you're going to get. Now, these two people, they can't convey that. There's no model, a piece of paper where they draw that line or anything down there. They're talking past each other. We also are proposing to involve stakeholders, right? Why do we do that? We increase transparency. People understand how decisions are made, right? We can identify concerns among people, among the stakeholders, among users. What do they care about? What are they worried about? We can identify broadly supported strategies, right? Creative strategies for achieving the objectives we want to achieve. then we can minimize conflicts, resolve conflicts. If we keep that discussion going with the stakeholders, and we can build public support for what is being done. So structured decision-making is not the traditional research and management relationship, right? What happens is managers have a problem, they discuss it with researchers leave, they go away, they do the research, and they just hand off the results to the manager and say, good luck implementing that based on what I found. Right? That's not very satisfying. That's not how I do my program, and I don't find that very satisfying at all. As opposed to the research management relationship under structured decision-making approach, right? First of all, research is driven by informing the decision, right? The focus is the decision, the MacGuffin, the decision, right? It's a joint effort, right? It's tailored to the problem at hand. And then the manager say, hey, you've got to incorporate some of these realities, some constraints that are on us, things that we need to consider, right? So when we talk about decision-making and learning, iteratively learning about how the system works, reducing the uncertainties, these things we're not sure about, right? That's just it focused at improving decision-making, not just gaining knowledge for knowledge's sake, gaining knowledge to improve the future decision-making. So I'd like to say that structured decision-making, the collaboration within it is a win-win, right? Managers, they can build technical capacity, right? They can get this expertise in these quantitative approaches and other approaches to help them with their jobs and improve their decision-making. The researchers, well, for us, science is relevant, right? Yay, people are actually using my science, right? The applied problem actually gets applied. And then it leads to more effective delivery or conservation delivery, right? It makes us better at making decisions. So I'm often asked, well, what does this work? This is pie in the sky. Well, I have dozens of examples if you want, but the closest one to what you all are talking about is I've been working with the Central Valley Project Improvement Act folks, the fisheries group there, for anadromous fish restoration since 2013. And we've implemented the structured decision-making approach in 3 phases. Phase 1 is what we call this rapid prototyping, which just involved a small group of folks from the state and federal agencies to figure out if that's what they wanted to do. Do they want to go down this path? Phase 2, we opened it up to the public. We got broad public support. And we have lots of folks that continue to come here. So ProTroll, you guys know ProTroll? That's Dick Poole. Dick Poole is a member of our science integration team. He comes to just about every meeting prior to COVID, right? And then John McManus with Golden Gate Salmon Association, he comes to our meetings, he participates. And if they have a difficult time expressing a scientific idea, we help them with it. I sat down with Dick and wrote a proposal for including Lake Fall Run into the bag of objectives that we wanted to have and build a model behind that. That's work with the stakeholder group to try to implement some of their ideas. And then this last phase, we actually came up with a near-term restoration strategy. And this is something that Reclamation and Fish and Wildlife Service are implementing right now to the tune of $80 million. We have this process, priorities for research, priorities for monitoring, and Reclamation through their office have already had $80 million to obligate. They've obligated already $40 million, and password additional $40 million for this next part of the restoration strategy. So it's real. This is a real thing. And then due to our success here, Reclamation has asked me to initiate a process in Klamath Basin this year. So we'll see how that goes. Guys, I hope you saw the email from Rod Whitler with Reclamation, who I've been working with since 2013. So this is a real thing. This is something they actually use, and they're asking more of us, the science integration team. OHRC benefit from this, right? Well, it's an inclusive collaborative process, right? We're involving other parts of ODF&W in this. Melanie and I already have work going on with the RED team on coastal salmon management, particular habitat work that we're working on. We can leverage that. We can make this a bigger thing, a better thing, right? You'll get science-based decision support. This is to prioritize any of your activities, management, research, monitoring, right? And we can leverage ODF and W research and monitoring, these other things. So our budget goes beyond your budget, your limitations by far, because that's really what I think it takes to implement the process. However, an alternative would be to go through this rapid prototyping process. When you complete the structured decision-making process relatively short time, right? We would take you folks, the board, maybe a few other folks outside, and go through it in about 6 months' time to see if it's something that you think is useful to you. Roughly about 6 months of a postdoc time to do that. So it'd be part of that, and no 50% overhead with us at the unit here. So we could do this to see if this works for you. Is this something that works for you? Is it something that you think is worth pursuing, etc.? So any questions?
Do we have questions from board members for Dr. Peterson?
Yeah, Jim, that's Steve Jacobs. I had a question. It just seems to me that this is more appropriate for beyond the OHRC, you know, in terms of the entire, well, Department of Fish and Wildlife, in terms of their approach and objectives for management. The OHRC fits within that to carry out specific science. And this could guide the OHRC in the long term or the bigger picture, but it just seems to be beyond kind of what our mission is in based on what I'm hearing from you.
We can constrain the scope of this, I think, and I think, and then see if it's working for you. That's basically it, Steve. You can reframe the decision space if you want to. But I think that OHRC, if anything like this ever gets implemented, they have to be on board, right? We need hatchery management is key to salmon management. You can't leave it out of the picture. So it would have to be part of the process. So where do we start, Steve? I think this is a good place to start.
And what kind of input? How do you seed it with input data? That's another thing I'm confused about. Or do we have? It seems like we'd have a lot of uncertainty in what we'd have as far as input parameters or knowledge for input.
You guys are actually doing pretty good. ODF and W does a wonderful job with monitoring, collecting data. The sort of stuff we started off with rapid prototyping and the CDPIA, just about 2/3 of what we did was based on expert judgment for the rapid prototyping. But then we started adding data to it, right? It was a placeholder in the beginning. Then we started adding data. And there's a lot of data that's being collected in coastal Oregon by ODF and W, not mentioned the Willamette. And I got the opportunity to use some of the data that was collected in the Willamette to create probably what I think was the best empirically supported decision model that I've ever made. And that was the Willamette streamflow Chinook salmon model that I created. And that was awesome data.
Yeah, I did notice that you were going to use Chinook as your species to prioritize, which I thought was curious because as far as the hatchery role on the coast, Chinook, the hatchery involvement with Chinook is far less than it is like, for example, steelhead. We have a lot of basins that don't have hatchery programs for Chinook, but steelhead are more widespread. So I thought that was curious.
You know, we can, whatever your objectives are, that's what it is. We give an example of, yeah, sure, we can work on Chinook, but that's because that's what we happen to be working on, primarily salmon, not steelhead necessarily with the red team.
Yeah.
Okay.
May I ask one quick question? I know we're time constrained, Scott, and just last thing, your budget is kind of above our range as it is anyways, but it sounds like if it were to be applicable to OHRC, we'd probably need a more constrained decision space. What what kind of time frame would it take you to turn around a revised, more boxed in proposal that would act more as a pilot, I guess, to the point that Steve made? You know,
Morning. Is that good, or do you want one? I can get you probably this afternoon too. Kind of depends on how long this goes, and I've got a couple other things I need to.
No, that's reasonable. I think we'll, you know, take that into account in our deliberations, and then if we have an interest in looking at a revised box around that, we'll definitely let you know.
Okay, thank you. Moving on to Craig Lawson.
All right, can everybody see and hear me so far?
Yes.
Okay, excellent. Good morning, everyone. My name is Craig Lawson. I am the senior hatchery technician here at the Oregon Hatchery Research Center. And the project that I will be presenting for you today is a survey and evaluation of ODFW's angler-caught broodstock programs for coastal steelhead. I work for ODFW, obviously, and I work at the Oregon Hatchery Research Center. And my collaborator on this project is Mark Johnson, who works with ODFW and Oregon State University. So we'll dive right in and look at the generalized research question here. This is actually a more refined version of the research question that members of the team here at the OHRC asked after the biter study, after we viewed the presentation on the biter study. And the main focus of this particular research question is to assess the impacts of angler-caught broodstock ACB program methods and materials on the fish that are utilized in these programs. The images that are pictured on this slide are examples of the methods and materials that are being used. And I'll go into a little bit more detail about those in later slides. To give you a bit of background on angler-caught broodstock programs, there are currently 8 ODFW hatcheries that have ACB programs for steelhead. And these programs have historically varied in their success at production of offspring from angler-caught broodstock. And what we want here at ODFW is to follow the 4 goals that are set out in the ODFW mission statement to the best of our ability with angler-caught broodstock programs. To summarize those goals pretty quickly, one is to foster fishing opportunities, to emphasize the hatchery role and sustainability of native populations, to maintain genetic diversity of wild populations, and to minimize the ecological impacts caused by hatcheries. All of these are very relevant to this research question and the study of angler-caught broodstock programs within ODFW. We believe strongly that this project follows the mission of the OHRC to produce research that attempts to understand the impacts of hatchery practices and to enhance these practices in a manner that's consistent with ODFW goals. Additionally, the emphasis today is on projects that really emphasize fish production for fisheries. This project qualifies for both of those sort of subtopics of fisheries and fish production as it relates to angler activity and fish production methods. So it's sort of a 1-2 punch with this project. To go into the approach that we are proposing, generally the project can be divided into 2 overarching parts for this process. The first being the program analysis, which can further be divided itself into 2 major approaches. The main components of the program analysis are the data request, which for this project, we're looking at angler-caught broodstock information from 2015 to the present, and that's sort of necessary to limit the amount of data that we can get on this as a starting point. The sources for this data will be the hatcheries where the broodstock and broodstock spawning is located, and the district biologist's office, so that we can get more generalized program data regarding each angler-caught stock. Information that we're going to collect includes collection location for the broodstock, the date of collection, spawning date, average holding time. These are just examples. We're looking to collect all of the information that's even potentially available regarding these programs. And then from the district biologist's office, we're going to get the more generalized information like program letters describing this information or describing these programs to the public, and getting a better idea of the actual angler pool that are participating in these programs. The second part of the program analysis is the site visit. At the site visit, we proposed to photograph infrastructure, talk through the collection and holding process with managers and crew that work at the hatcheries where these programs are located. That's really essential to gauge understanding and to gauge practical analysis of the methodologies and equipment that's being used. We're going to be looking at system design as well at these hatcheries, because each hatchery is going to be very unique in terms of its water source, in terms of its infrastructure. And then one last thing to mention on the site visits, 2 hatchery data requests and 2 site visits are complete. So Alsea Hatchery and Cedar Creek have already been forthcoming with their data on the angler-caught broodstock programs that are occurring at each of these hatcheries. And I've visited both hatcheries and gone through the infrastructure survey as well. So the second major overarching approach for this project is the perception survey that's going to occur concurrently with the program analysis. Went through a variety of different types of surveys, but we landed on SurveyMonkey. So we're going to use the SurveyMonkey software to produce a survey that's going to be sent to hatchery managers. Now, the topics for this survey are going to include public awareness and engagement, perception and attainability of program goals. That's a big part. And then the thoughts on the potential for expansion or reduction of angler-caught broodstock programs. Long-term, we're hoping to collaborate with OSU science communication experts to get the most out of this assessment and see how it can be expanded to gauge public perspective regarding these programs. And the first iteration of this survey is ready to be sent to hatchery managers of ACB programs. I actually just got word this morning that there has been movement on that front and that I've been integrated into the SurveyMonkey license. So I will be able to send that out later today, possibly. So the next portion of this is how will this information be used? And there are a variety of use cases that can be gained from the collection of this information. The first would be to gain insight into the perceptions of ACB programs from both the public and members of the agency, ODFW, the involved agency itself. Developing a better understanding of ACB programs and their applications and addressing the impacts of methods on the collected fish. Those 2 use cases will hopefully be used to further the second to last goal, which is development of best management practices for ACB programs. If we can develop a strong unifying methodology for the incorporation of ACB programs for steelhead, that provides sort of a foundation for the potential building out of further ACB programs, either just on steelhead or sort of expanding into other species as well, potentially. And then finally, that plays nicely into providing a source of research questions regarding ACB topics. When we initially started talking about this project, there were a variety of topics that could have spawned just from this one research question. So we're looking to determine what we know and what we don't know about angler-caught broodstock programs, and to expand on that knowledge to further research with ODFW and potentially with OHRC as well. Just really quickly to go over budget justification a little bit. Pictured is the budget from the proposal. A few notable points are that 2 site visits to hatcheries have already been done at cost that's well under the projected rates. The data collection in terms of the data request has also been undertaken for 2 hatcheries as well. So that's approaching 25% of the potential data collection that's already been undertaken with respect to the actual physical data request. ODFW has also purchased licenses for SurveyMonkey, and that is at well under projected costs to the OHRC. So looking at all of that is very interesting to consider with respect for the already quite small budget for this project. And at this point in the data collection process, so far, this project has proven to be relatively low cost, and the potential for the data yield from it is relatively high. So that's a very generalized overview of the project. Just to go over some concluding remarks here, there's a range of potential benefits that ACB programs provide to riverine systems in general. And developing our understanding as an agency of ACB programs is going to increase their value to the public. Why research them further? Every hatchery is going to be vastly different from other hatcheries. They're going to have different infrastructure, history with these programs. A great example is some of the stuff that I presented on in the previous slides, mainly with the research question. Those were examples of holding system differences that occur at Cedar Creek and Alsea hatcheries. Just between those two hatcheries, the methodologies that they use for their programs are vastly different. And we're definitely seeing in the data that there are historical differences in success that so far have correlated with changes in their methodologies and changes in their infrastructure programs as well. We don't yet know, or we don't yet have a firm grasp on all of the different factors that are going to impact an angler-caught broodstock program, but we do know that a reduction of stressors is very important for the survival to spawning success of the angler-caught broodstock. So developing methods for reduction of stressors across the board is going to be definitely an increase of value to these programs. And since the public makes up a very integral part of ACB programs, ODFW's understanding of how they perceive these programs is going to be very important in increasing these programs' value to the public generally. That is all I had for this presentation. Thank you for your attention. And does anybody have any questions regarding this project?
No further, excuse me, no further questions. I guess we'll move on to, is it Andy Dittman going to do this or Carl?
Mr. Chair, we do have a minute or two if, according to the schedule, if there are questions from the board for Craig.
Oh, there are? Okay. Now, sorry, go ahead.
Well, it's just that we do have a minute. We're a bit ahead of schedule. So if there are questions from the board, I think it could serve the board well to.
Yeah, I had a quick question, and maybe either Craig or Mark could chime in. Is there anything that has come up, I guess, as you've thought about this small piece of work to augment, the prior work that, has sort of triggered any additional thought subsequent to the submission, or do you feel like this is, kind of moves the ball down the field sufficiently based on what you've laid out? Anything else that sort of come to mind while you formulated this?
So the more I've looked into this topic, definitely the more questions I've had, and that's kind of why we sort of instituted the 5-year limitation to start off with on our data collection, was just because if we expand past this, it's probably good to produce something, was our thinking,
right,
that actually had data value before we moved on and sort of assessed further what the other factors might be. In terms of questions, yeah, I've got pages and pages of further questions regarding this topic, but I'm trying to sort of focus attention for a little bit to see if we can produce something.
No, that's good. I think this is a very focused effort, and appreciate that. I may have some other questions for you as sidebar later around this and the follow-ons. Appreciate that. Thank you.
Sounds great. Yeah, be happy to answer them.
And I had a quick question for you. So as far as the end products from this exercise, so you're going to compare different the different methodologies or techniques that are used across different hatcheries for angler-caught broodstock, and are you and just come out with a range of what those differences are, or are you actually going to have some metric that's going to compare the relative success of one method versus another method across the different hatcheries or programs?
So our main variable of focus right now is broodstock survivorship to spawning. So we're going to compare the hatcheries against each other in terms of broodstock survivorship to spawning, and then we're going to compare the hatcheries to themselves historically within these 5-year, this sort of 5-year cohort. Further aspects of the project would expand that range further. But if there are market differences within the hatchery comparison to itself historically, we'd like to go back and be able to point to something and say, okay, well, they installed a new holding system for their angler caught brood stock that had a variety of different features to it. And next year they saw a 15% reduction in mortality of their angler caught brood. So if there are a variety of these potential correlations, then we can go back further and attempt to sort of statistically explain that.
So you may come up with recommendations for best practices out of this potentially?
Yes, that is a potential eventuality for it. Within this particular study, we're not looking to come out with something that says like, this is what needs to change now. But if we can sort of, it's almost a pilot study that provides a use case for potentially creating a best management practice.
Okay, thanks.
And if I could just briefly round out Craig's answer here, and then we do need to keep on schedule. But I'll just briefly add that this work is a follow-up that was recommended by ODFW's Fish Division in response to the results that we saw from the OHRC funded biter studies. From that study, we saw that angler caught broodstock produced half as many adult returns as captive broodstock. But before taking that information and making changes to programs, Fish Division and leadership felt that it would be important to see if there might be differences among programs in the way that they collected the broodstock. We heard back that, from some programs, look, our program's working really well. The results that you have there from the LC don't apply to us. And so this is an effort to address that issue and question, and then develop best management practices, just as you indicated there, Steve. So. Mr. Chair,
thank you.
Dr.
We got to keep moving on here. So, Carl, are you going to do this next one?
I am. This is going to be very fast. Last time I spoke about the Elk River project, and so I'm not going to repeat what I told you about 2 weeks ago. Basically, what we need is about $20,000 to buy the amino acids that are needed to scent the Elk River fish ladder for next fall's return. The reason that we don't have that in the budget right now is really threefold. About the time when I wound up being the PI of this project, the budget was cut pretty drastically by ODF&W, and it basically eliminated any wiggle room that one always builds into budgets for events that happen down the road. And what's happened down the road since we proposed the original budget is a couple of years of inflation due to the pandemic, and the cost of amino acids just skyrocketed unbelievably. And then secondly, when we originally planned the project, we were planning on 2 months worth of scenting during the adult return run. That would have been last fall and then this coming fall. But in reality, fish kept coming through the new year. So we actually last year scented for 3 months, but the budget had money in it only for 2 months. And so this coming fall, we also expect to have to scent for 3 months. So we basically have a shortfall of amino acids for 2 months worth of scenting the fish ladder. The budget amount was calculated roughly around the first of the year when we realized what was going on. And so this is the new quote from the vendor of the amino acids. All we're asking for is money to purchase the amino acids. There is no overhead on this money. And the money goes to OSU to purchase the amino acids for use at the Ohrc River Hatchery. So if there are any questions, I'll be pleased to tackle them.
And Dr. Schreck, Dr. Andy Dittman prepared a couple of slides. Would you like, which really just reiterate the points that you've made?
Do we need to show them? I've not seen them, Mark. I've seen they just came last night.
I think you covered it quite well, Carl. I think we're good. If anybody wants to see them, we're welcome to share them, but I think Carl covered our points. Thank you.
I have a question that it was part of the program was to find a odorant that was readily available, non-toxic, and affordable. Is this gone? Is this increase in price kind of due to inflation and this whole supply chain business, and you anticipate it to go back back to where it was?
No, not at all. No, the original design of the project, the original long-term goal is to do exactly what you said, Scott, find a readily available, inexpensive product to use. But first we had to proof of concept to really make sure we could do this. And so that's why the cost here, for this first part of the study. So in the present work that we're undertaking in the lab is to do exactly what you're doing, coming up with alternatives. So the scenting of the ladder now is a proof of concept rather than the final product that we hope would be used.
Thank you. Any other questions for Carl or Andy? If not, we'll move on to Amy Reed.
Hi, can you hear me okay? I have some slides prepared for you. I know I've spoken with you before. Thank you all for taking some time away from your day to hear about our projects and what we have to say. Mark, can I share my screen here?
You should be able to. I've made you co-host, so there we go.
Great. Well, thank you all. If you hear feedback from my screen, it's because my computer fan is working really, really hard to keep up. I'm just going to talk really briefly about a little project that we have that we'd like to continue with. You've heard me talk before about thiamine deficiency, so this is along the same lines. Our project is efficacy of injectable thiamine treatments to alleviate deficient thiamine levels in eggs of steelhead trout. And my interest is really in steelhead trout. Sorry if you haven't met me before, I'm Amy Reed. I'm the Senior Fish Health Specialist at Oregon Department of Fish and Wildlife and for Fish Health Services. I'm not going to give much of an introduction to thiamine deficiency complex. I spoke to the OHRC board a few months ago, and we had a really lovely conversation about it. But just to summarize quickly, thiamine deficiency complex is a syndrome in which thiamine is not available in the eggs for the developing fry to take advantage of, and they end up with neurologic deficiencies, failure to thrive, and death. And so mortality can be quite exquisite. If you don't die from thiamine deficiency as a fry, you can grow up and have significant impacts to your health. The research has shown that the fry will grow up to have increased oxidative stress, lowered ability of white blood cells to attack and kill bacteria, as well as lowered antibody production. Of course, it's caused by a couple of things that we're looking into, but mainly it's the high thiaminase diet for broodstock, where the thiamine does not transfer into the eggs. If you are a thiamine deficient fry, there's a couple of ways that we can get thiamine into you that clinically, from a clinical perspective. And one of them is to inject thiamine hydrochloride into the broodstock so that the thiamine has between 7 and 14 days to work through the female's system and get developed into the eggs so that the eggs have the thiamine on board before fertilization and before spawn. The second is to supplement the eggs or fry with a bath of thiamine mononitrate. And that bath is water soluble, so it just goes across a soft shell of an unfertilized or just fertilized egg, or it can absorb across the tissues of a newly hatched fry or alevin. And we have detected thiamine deficiency in the steelhead from Oregon, in the fish that were collected in 2018. We had them analyzed in 2019 at a USGS facility in Missouri, and were able to detect that the levels of thiamine were not sufficient for these fish to thrive. And one of the major questions that we have is this: is there an easy and affordable treatment that could be normal for steelhead production in Oregon that can help to, number one, decrease mortality in steelhead fry and have our survivability rates increase? And then the second is to allow our steelhead production to be a healthier group of fish for them to be more immune competent and more robust, and hopefully not succumb to as much disease. So this is a little project where we've done a small pilot project before where we're supplementing eggs at fertilization with that thiamine bath. But this project is to say, well, do steelhead adults respond to an to an injection of thiamine before spawning? So what this proposal is how is that going backwards? Here we go. We have, what we'd like to do is to supplement thiamine in a couple of different ways for steelhead eggs, both an injection and an immersion. And so the design is to take 30 winter steelhead adults, inject half of them with thiamine, and then mock treat, not inject the other half, so that they're still handled and tagged. And then each of those groups, we would have the eggs, half of the eggs from that female would be treated with the bath, half of them would not be treated with the bath. And so really, here's what that looks like when we did a pilot project last year. We take the eggs from the female and split them 50-50. Then we would treat those eggs with thiamine mononitrate in a bath for an hour at 1,000 parts per million. After that, they get rinsed and then trayed down and then water hardened with iodophor. That essentially would give us 4 egg groups or 4 fry groups once they hatch. Those would be fish that got double treated with thiamine, injected and immersion, fish that only got treated once by injection, fry that got treated just once by immersion and no injection, and then a double negative, this no thiamine treatment control group. So we would have 4 groups of thiamine here. And then these fish, these eggs we would like to rear at the OHRC and raise them with all identical conditions of normal steelhead hatchery rearing. Surface water that's ambient temperature in late winter, early spring. We would disinfect them with iodophor at fertilization and treat them 3 times a week with formalin to control for fungus, which is really standard of care for eggs in that facility. So these eggs then would be trayed down and able to rear in these 4 different groups. Our questions are, does thiamine supplementation and method of delivery affect fry survivability? There is research to say that eggs that get thiamine at the very beginning before spawning end up more immune competent than fry that are rescued after hatch with thiamine bath. And that's what we're looking into for steelhead. So that has been investigated in other salmoniid species. How we're going to assess these questions is by measuring both egg survival and fry survival at 2 different times, both at the egg shock at eye up and at ponding at swim up. And so this would tell me 2 different things. Were the eggs able to survive? And then were the fry competent enough to utilize their yolk and get on to first feed? We have a lot of issues with dropout disease and eggs, with steelhead fry, with pinheads that get so skinny that they just never go on to their first feed and perish. So we'd like to take this experiment out to about 12 weeks so that we can measure fry survivability after about 12 weeks of going onto their feed. There are some curious questions here that we'd also like to ask if we can get to it, would be are egg size or fry size different with treated and untreated groups and with injected versus immersion groups? Do we have any difference in presentation of coagulated yolk, which we see very often in steelhead in Oregon? And can we monitor for infectious disease outbreaks? Is there a potential for one group to break with disease, infectious bacterial disease, when another group doesn't? Overall, what we'd like to do is to inform standard of care for steelhead rearing in Oregon. And I think that this could be a really practical way to help us determine which methods can be best at delivering thiamine to our steelhead for production. The second part of our assessment is to measure the egg thiamine content in all test groups to assure that the thiamine is being taken up and available to the egg. but this is a major problem in a lot of my thiamine questions. And there's many, many, many questions from many groups in Oregon and ODFW and USGS and NOAA and OSU about thiamine. The way that we can answer them is to analyze thiamine in the eggs, but there's not a lot of ways to do that. I got my initial thiamine analysis at a USGS lab in Missouri with some experts there who have studied thiamine their whole careers. That's using a fluorometric analysis, and they're developing a liquid chromatography mass spec, which is a gold standard, which would be excellent. The folks at USGS are retiring in 4 days, so all of his expertise is going with him. I'm hoping that we'll be able to continue to work with them. The other way that we can analyze eggs is at a SUNY lab, and that's HPLC method, and those methods are published. So the question we're asking is, can we develop this testing HPLC at Oregon State University? So I'm asking this question with Linus Pauling Institute and Department of Microbiology. We're trying to see if we can treat our eggs in this spring and January, and we've taken these eggs and analyze the thiamine. Can we do that here at the Linus Pauling Institute at Oregon State University? And so that's the second part of what we're trying to do here. A part of my budget includes development of the assay and use of HPLC machines at Linus Pauling Institute. And I've had some preliminary chats with them of their willingness and availability to do that, which is warm. And the rest of my budget is supplies. We have some time being volunteered by the hatchery staff at ALC, as well as Jen Krichek, of course, has been helping us with this as well. And so we're requesting just some money to keep this going. And then Chris Suffrage has been really wonderful. He studies thiamine in environmental systems, and he has been very excited to help me on this project and see if we can develop some thiamine analysis at OSU and potentially Linus Pauling Institute as well. That's just a really quick summary of what we're working on and what we're asking for this little project here. I'm happy to answer any questions you have.
Yeah, I had a quick question. How much of an issue is fry mortality in steelhead rearing in Oregon coastal hatcheries?
Yeah, so we have, that's an interesting question. We have some programs that see between 12 and 15% loss at shock, at egg shock. And then we have another maybe 10 to 13% loss at ponding. There are some programs that will plan. I mean, we've known that steelhead rearing in Oregon, that steelhead are just really finicky, and the babies, you know, you have to get them going. But when they're this teeny tiny babies, they are so delicate and they just don't well. So we end up usually taking a certain percentage more than we know we need. So we'll say, oh, we'll take 30% more eggs than we need for production because we're anticipating 30% fry loss. And that's wild. That's wild to me, because what that means is that these fish are being raised in higher densities because we are expecting more, you know, we need to take more of them in order to meet production. But also, that's not a terribly ethical way to move forward. So we're trying to figure out how to mitigate that. But steelhead in general, just they are a tough cookie to get going when they're that little.
Okay, that puts it in context. I appreciate that.
Sure. Well, thank you all.
Any more questions for anybody? We've got about 5 minutes ahead of time or so. So if there's any other questions back from some of the other presentations, go ahead, ask questions. We've got a little bit more time.
Thank you, Dr. Reed.
So if there's, next we have coming up a break we can take a few minutes early, give us a little more time later. So the presenters can stay and listen for the discussion, or you're welcome to go do something maybe more productive if you'd like. So with that, is that okay, Mark? Does that work if we take a 5-minute break right now? So it's about 10:45. We come back at about 10:50 and we'll begin the discussion. Does that work for everybody?
Uh,
Go ahead.
Yep. No, that works for me. Sorry. I had
No, I think that sounds like a fine plan, Mr. Chair. And maybe before we break, just briefly, I'll remind board members, if you look in your chat, you'll see a link to a poll that you can use to communicate your preference and priorities for the proposals that you've heard today. This, of course, does not constitute a vote. It's only a tool that you can use to communicate amongst yourselves. And I'll put that up on the screen when we come back and discuss the proposals that you've heard today. Then I look forward to hearing your recommendations in the form of a motion if you see fit to do that. Thank you.
Okay, thanks. With that, we'll take a 5-minute break. Be back at, I got 10:47, I guess, 10:52 or something like that. Okay, thank you.
And maybe I'll ask here, are there any board members here in this moment that would like to use the poll but have not been able to access it for one reason or another?
You want to show that poll, Mark?
Yeah, I can do that now. Okay, you should be able to see the poll now with results. And I'll remind, as the discussion proceeds, board members can change their answers. So if someone is convinced or compelled to do that, the dots can be moved around. And this is just a tool to help you all see where favor is landing at present.
Okay, yeah, if you can take that down, Mark.
Oh, okay. And I can, we can revisit it at any time.
Well, yeah, you can leave it there. Okay, I've been trying to think how we're going to get through this discussion because we've got about 4 minutes or so on each one. So I think it'd be easier to take them kind of out of order and get the easiest ones first. I mean, it's like a no-brainer. We need to fund the odorant, the $20,000 for that. Another one that, well, let me see. The rest of them are pretty much even, but that one, yeah, that one's like a no-brainer. So I'll give you then my opinion of some of them. I guess we could do it one at a time. I'll give you my opinion just to begin the discussion. Like I say, I don't think there's going to be a lot of discussion on Carl Schreck's needing that deodorant.
Yeah.
That's pretty positive. So,
Mr. Chairman, can I ask a quick question before we dive in, all because I think it's germane to how we think about these because they're broadly divergent on budget. What happens if we only, and maybe it's to both you and Mark, what happens if we only allocate a portion of the budget? Is it a use it or lose it type circumstance, or does that roll over for us into other periods?
we can partially fund them if we want, or say we'll fund some part of it if they get the matching funds, or the rest somewhere else. If they can't get it, we
That wasn't the question. Actually, it was more about our budget. If we say, for example, if we allocated 44,000 out of the 144,000, what happens to the other 100K? Does that roll into our ongoing budget, or does it go away?
It does not go away. It carry over.
Okay.
Even
Thank
into
you.
the next biennium.
Thank you.
Even by statute, the carryover 10th year goes to back into hatchery improvement.
Actually,
anything else?
Mr. Chair, I believe that legislation.
All I do is tap on it.
Excuse me?
That legislation was amended so that unallocated funds roll over into the next biennium. They can carry forward.
Yes. Yes. But no, we don't need to discuss it further. Okay. The other one that, say, I'm not a scientist, but I asked some folks around what they thought of this Amy study on the thymine, and that seems to make a lot of sense. It's not very expensive, and we could probably get some really good results out of it. So I would certainly be in favor of that one. So any discussion on that? Is everybody in favor of supporting that? $13,000, not very expensive, and it seems like a pretty good return on investment to me. Tom, any thoughts? You're muted.
This is Carl. I agree with you, Scott. That makes sense.
But
I support that. This is Steve Jacobs. I think it's efficient, cost-wise, and worthwhile.
I'll take this opportunity to encourage the board to generate recommendations through passage of motions, if you're ready to do that.
Well, I thought we'd combine a couple maybe in a motion and then debate some others. That's not a problem. So it looks like we have favor for the odorant 20,000 and for reeds 13,000 or so. Other one that could be fairly easy, that is the Lawson, the $3,700. I'm kind of impartial on that, but I'd like to hear what people think.
This is Carl again. I think that would be money well spent. I mean, it's extremely modest, but it, you know, there's, like Craig was saying, there's other questions that could be asked. How do we improve it? But it'd sure be nice to, you know, start out at in some common ground of understanding what the differences are between places.
Yeah, return on investment's pretty good, I would think. Ted? You're muted.
Yeah, this
Can't hear you,
is
Ted.
Pete. This is Pete Grundy. I have a couple of questions on a couple of those. So for Craig, can you just tell me the number of angler caught broodstock programs that are currently taking place in Oregon, and does the evaluation of those programs include all of the programs that are currently in place in Oregon?
That question
Mark?
just directed at Craig.
There we go.
That's a question for Craig, yes.
Yes, Craig is not in the office at the moment. I
Or
believe,
maybe Mark, you could.
I believe he said it was 8.
I believe it is something like 8. And yes, the proposal is to survey all of those programs.
Okay, great. And then one other question I had for Dr. Reed is that you mentioned in the timeline for the treatment of the steelhead eggs that, you know, that would be undertaken in this brood season. And so have those eggs currently been treated at this point then?
Yeah, we took advantage of some of the timing of the steelhead spawn and collected some eggs and treated them just a few weeks ago to hopefully get them going in the hatch house here. So yes, those have already been taken in and the treatments have been applied. It's also something where we might be able to repeat it since it's still within spawning season for steelhead.
Okay, great, thanks.
Is there any other discussion on these three? The odorant and the thymine and the angler caught? If there's no further discussion and people are in agreement, I'd entertain a motion to fund these three projects at the budget that was submitted to us.
I'd make that motion.
Okay, let me see. Looking for my budget thing. So we have a motion to support the Amy Reed's project for $13,000. Carl Schreck, or the odorant, at $20,000, and Craig Lawson's angler caught at $37.66. We have a motion to support those three projects and fund them. Do we have a second? Have a second on that?
Second.
We have a second, and who was the second? Was that
Steve?
Steve? Okay. Peter made the motion. Are you keeping track of this, Mark? Yeah. Mark, Terry, keeping track of who's for the motions? Okay. Is there any further discussion?
We're recording the meeting, Scott, as well.
All right, thank you. Is there any further discussion on these projects in this motion?
Yeah, Scott. So does that leave us with about 75,000 unallocated then?
More than that. It's so basically we're at 37,000 spend roughly on a 144 pot.
Right.
So I think
Oh, okay. I got the
107,000 left.
Leaves
okay.
over at that. Yeah.
Okay, I got the numbers off.
I could benefit from a clarification of the motion. Which projects were recommended in that motion?
The last three on the list, so it would be the angler caught, the deodorant, and the thiamine deficiency. Those three.
Yeah, and we can augment for the public record for clarity as Lawson, Shrek, and Reed were the presenters.
Okay.
Thank you. So that's quite right. That leaves nearly $100,000 of unallocated funds with if that motion were to pass at this point.
Okay, we have the motion, we have the second. Any further discussion? If not, I don't know, how do you want to do this vote, Mark?
I believe we could do a roll call vote if that pleases.
Would you do that, please?
Yeah, just one moment. I need to. While I get this ready, if there's any further discussion by the board.
Yeah, this is Derek. So basically, we'd possibly move forward funding these three, and then look at what are over $100,000 left. Try to make the big decision of where that goes. Am I on the right track?
Yes.
Yeah.
Where it goes,
And the other
keep it. Yeah.
The other context is that only 2 out of those other projects of 4 would have been able to be fully funded if we used the higher allocation, and 2 would need significant revisions in order to be considered, for funding from us, unless they had other funding sources.
Right.
Okay.
Okay, so I have our rule in front of attendance here in front of me. Would you like me to conduct a roll call vote, Mr.
Please.
Chair?
Yes. Yeah, go ahead.
And everyone is content with parliamentary procedure here in this moment? Okay.
Yeah, absolutely.
Okay, so the motion is to make a recommendation of support for projects proposed by Lawson, Shrek, and Reed. Working, is Dwight Collins with us today? Okay, I don't believe so. Derek Wall.
Yes.
Seth Eisenberg.
Aye.
Tom Alcare. Tom?
Yes.
Okay, thank you. Jack Smith is not with us today. I believe. Lindsay Ball.
Mark, this is Kathleen. He had to leave the meeting early, he said in the chat, I believe.
Okay, I missed that.
I figured that you guys have been busy.
Thank you, Kathleen. Ted Simon.
Yes.
Chuck, I know, is traveling. Scott Starkey.
Aye.
Steve Jacobs.
Yes.
Pete Grundeck.
Yes.
Travis Mackey. Travis. Travis, there you go.
Yes, aye.
Thank you. And those are all voting members. By my count, this was unanimous passage of the motion to support proposals to be led by research to be led by Lawson, Schreck, and Reed. Chair, I believe it's
motion
to declare passage of the motion.
Eyes are in favor. The motion passes. Thank you.
Right.
Okay, moving on. Say I'll throw in my 2 cents worth and then open for discussion. The Ballsy model. I'm not too much in favor of models. It looks like nobody else is, but either. So there's probably not much discussion there. I'm just concerned with models mainly that they're only as good as the information that goes in or the bias that goes in. And there seems to be plenty of bias around these hatchery wild issues. So kind of what goes in is what comes out. And so I get pretty nervous about the models, and it looks like everybody else is pretty much in agreement with me. So if there's no further discussion, I guess, or with the more discussion, the Ballsy is out for and we'll move on to the, let's take a look at Peterson after that. Is there any more comments on Ballsys?
I did have a question on that. I mean, I think modeling approaches can be problematic to your point, but they can also be illustrative, especially about guiding future planning. I'm not sure that's the right one for us. I mean, the other challenge is that now that we have a budget left of $107,234, if that could be significantly, slimmed down to the point where it would be viable, and it doesn't seem like there's enough board interest to maybe warrant asking him to go through that level of revision. But I would want to put the caveat around the modeling, because I think it can be a very effective tool. But it does have, to your point, it's very governed by the inputs and also the parameters established in the model. But there's a high degree of transparency that you can provide on that. Doesn't seem to be applicable here, but I did want to add that as a piece of color.
Also, we need to keep in mind too that the funding is from the fishermen, and what are these going to, which of these are going to get the best return to the anglers who are footing the bill? Yeah. Well, so it doesn't look like there's any interest in that one, and not too much interest in Peterson. My input on that is, I don't know, about the same. Would be an interesting model, but pretty expensive and pretty easy to manipulate those things.
I think when Jim talks about, this is Carl, when Jim talks about models, I think he has something very different in mind than what it appears. We all operate with models in our brain, and what he's trying to do is get people to be explicit with each other as to what those models are so they can communicate. And he's been successful in doing that. And I know that I've served on recovery teams, and he's exactly right that people are usually talking past each other. They maybe want the same thing, but they're coming at it very differently. And so I think, when Jim says models, yes, you can put mathematics to this and it works, but it's really trying to get people to be clear on what is it they really want and what information is that based on. So that's where he's coming from. It's a very different use of the word model.
I hear you, Carl, and some of it makes sense, but just take for example the incubation box issue. There's a lot of people that are against incubation boxes, and a lot of people think they're a wonderful tool. And so you can get that input, but still it's the bias of the people that control the policy that is going to dictate what happens out of it, regardless of the input.
Yep.
In a lot of cases.
Yeah. I mean, I thought Steve made a very good point regarding Peterson's model on the integrated decision model. I think it is probably more broadly applicable in what he presented across ODFW. I think we'd be a little remiss by not providing him some guidance. I don't think we necessarily feel like the pilot is for us is the right approach, but it feels like in order for him to be able to get a toehold for that to be considered as a management tool, that a pilot frame or pilot framework might be applicable. And I guess my take would be I would like to see what he might envision with input from us if that were the case. And when that may not be our mission and that may be beyond it, but it feels like maybe some guidance would be warranted in a formal perspective, because I do think what he's presented is actually valuable for ODFW as a whole. I don't know if we're the entity to basically serve as a pilot organization, but I think that decision space that he mentioned that could be kind of winnowed down might be a framework. Is there anybody here that feels like that might be something that would be applicable for any of the issues we've been dealing with as a board? I don't think I'm have enough history here to be able to say what that would look like. I was intrigued by it though, and I think, Carl, your points are well made, that as a tool ends up being can be a pretty important one. I don't know if it's applicable to us, but I don't want to dismiss it out of hand.
Yeah, I think why Jim submitted the proposal is that it came out so highly rated out of the workshop. So the workshops collectively the workshop suggested this was a very important need. And it's basically to have conservationists and folks that are more pro-hatchery talking together and understanding each other. I don't know if Jim's still here. If he wants to move in.
Well, it doesn't appear there's enough interest in this one to go ahead with it. Let's review the first one, optimizing the rearing conditions. My take on that is it's really expensive. It's a good project, and there's maybe some good come out of it. This got microbe business I'm not all that familiar with, but it might come up with some really interesting results. But on the rearing process, there's been a lot of work already done on that. Carl himself has done an awful lot on the different rearing, so I don't know if how much more we're going to get out of it, or if that's the way the fishermen should be spending their money. But it is an interesting project. The more we can learn how to better rear fish, so it's a pretty positive thing. But I'm not sure I don't think that's one we should be funding at this point. And it looks like most people agree on that. Any other discussion?
Yeah, I have a comment on that. She did mention that there's an existing sample that she could analyze for the gut microbiome. And if she just did that, it would reduce the budget to $50K. And I think we should discuss the merits of that.
Yeah, basically what she has are fish-fed commercial hatchery diet, fish-fed a wild-like artificial diet, and fish from the wild. And so those samples could be analyzed. And that gets at the, the mission of understanding differences between hatchery and wild fish and how to make hatchery fish more wild-like.
Right. That's right, Carl. Yeah. But I think that has some, I think that has some that's applicable to our mission. And it's some new science that we haven't dealt with yet.
Well, this is Tom Elkire. I agree with that too, very much. I think it's a great idea.
Yeah, this is Travis. I tend to agree too. There's, like she mentioned, there's a lot of papers out there on, how the gut biome can affect how, different traits are expressed across different species. So, given the, the lower budget, I think this might be worthy of our consideration.
With the lower budget, did you say you still get kind of the output we might be looking for? I can't remember where we were with that.
I think that the output would be comparison of, can you achieve a wild-like gut biome with different cultural hatchery, different hatchery practices, and with the assumption that a natural gut biome is more like a desired trait as far as our hatchery rearing practices.
I'm happy to jump back in. I'm still, this is Claire. I'm still here if anybody wants me to jump in and answer any specific questions related to my proposal.
Did I capture that accurately, Claire?
So yeah, with the lower budget, it's we would be able to compare the effects of different feeding practices on the gut microbiome, and we would also be able to link that with effects on fish immunity, disease susceptibility, and growth, in addition to comparing with hatchery versus wild fish. Does that help?
Yes. I'm not the scientist in the bunch, so Carl and Steve, appreciate all your input.
I feel a little bit of a conflict of interest here because, Claire and I interact a fair amount. It seems to me like the state of the science is just exponentially growing, and where it is very comparable to, say, the stuff that Michael and Mike have been doing, looking directly at the genome, but a number of years ago, and we've seen, how productive that has been. So this is really breaking new ground, more so than using the techniques for how to get the data are well established, but actually what you can learn from it is really breaking new ground, kind of like genetics was maybe before the whole genome could be analyzed.
Well, I'm all for improved rearing conditions. The question, I guess, back to is this good expenditure of the fishermen's money right now? How would they prioritize it? So I'm kind of impartial on this. If there's any other discussion, or we could maybe let's put this one on hold and discuss Michael Banks' proposal.
So one real quick thing, Scott, a colleague of mine who's in Sweden indicated that out of all the proposals the EU gets on commercial aquaculture, the most common ones now involve the gut microbiome. It's been found that important with the aquaculture industry. So it is getting a lot of attention, but it's just so new that it's not in the literature yet.
Yeah, as far as bang to the angler, if we can get better survival, that translates to more harvest. So that's pretty direct application.
That's what they're coming from in Scandinavia.
Yeah, and I think also important, reduce disease transmission to wild fish is obviously an ongoing limitation for the release of hatchery fish as well. So it's another potential impact.
Yeah, and not only reduce the risk of disease to wild fish, but just disease within the hatchery itself. I know locally within Rock Creek, I mean, that was a big issue, and a lot of money was spent on, different filters and different medications to try to, keep those diseases at bay. So, I mean, I think there's definitely, if this pans out, they could potentially save money and, increase production.
Any other further discussion? If not, let's move to banks and then we'll come back to this. There seems to be enough interest that we're probably going to need to make a vote on this. Okay. As far as banks goes, the auto, to me, my opinion, the auto sampling is pretty good. As far as Michael's project that we've been funding, he needs to find a more efficient way to make the mate selection when the fish return. And in that context, I think it would be a great tool for him to use. And so far, it looks like there's been pretty good success with his project. I'm just going to throw out some skepticism I may have of the whole concept, is that it could probably be misused too. If fish can come up through a chute and get measured live, particularly, then somebody could make a decision: do we want to bonk them in the head or let them swim? And the problems could run into things like that. I was a little confused there. And you brought up, Steve, the question: is it just live fish or dead fish? And then it sounded like he was, potentially for both. Did somebody else pick up on that?
Well, I think initially for dead fish.
Right. I think he was only answering that the possibility would exist for using live ones if someone had an experimental design, because he's thinking about this for broader application than just his use at the Sandy Hatchery. I don't think he was indicating in his design that they were really headed towards using live fish, but I think that's what I took from his response.
And Dr. Banks is still with us, so if the board has questions for Dr. Banks.
I guess a couple of questions that I would have, Michael. How long does it take you to get a fin clip once you have the fish in hand anyway? Because you have to have the fish in hand to put it through the, through the machine. How long does it take you now to take a fin clip? And then the other question is, if the OHRC funds part of this, what's the likelihood of not getting the other part of it funded that you need to see this all the way through? And and when would this be? When would the other money be available if you need this in the fall already?
Sorry, it took a time to get to the mute button. Okay, I'm trying to get the questions straight.
Michael, how long does it take you just to get a fin clip once you have the fish in hand without the machine?
By hand, it's how long can you, snip a piece of fin and put it into a tube? It's pretty, it's about, I don't know, 30, 40, 50 seconds. But, in the hatchery context, when there are many, many other tasks at hand and you want to sample every particular one, and food share buses there and they're wanting to, load everything and go, it's just really hard. We sampled 1,300 over a few days, with Dave and I working both, and some of the hatchery members helping us as they could. But when we need to sample all of them, it's just not feasible. And that was impressed by Ryan Queen very, very vividly.
So your machine would allow all the fish then to be sampled?
Well, that's our hope. I mean, every fish is passed through this reader, and we would delay the reader a little, but you know, it's how quickly one can come up with robotics to do what we want to do is obviously something I can't say. I can do it. I haven't even been able to try yet. So, but that would be the goal. With regard to the funding, I'm keen to do this however I can. You know, as you already know, I already have a student that's paid for by other ways that is interested in this. I need a lot more support though than what that is. I have a proposal in more associated with the business side of things, but with this OSU Accelerate program, that I think they're making the decision on those funds. It's just 15,000, but they will make the decision on those funds this week. As I mentioned, disclosures about patenting, I'm working with OSU on that, and I believe that will make applications to SBIR more competitive. And I mean, I don't know the likelihood of those funding attempts. It's much more competitive in the national arena, but I understand there's a lot of money that this country puts behind supporting, taking innovations into the free market. So I'm just doing what I can, and I'm excited about realizing it in whatever way I can. And so that's my proposal to you.
Thank you.
My input then on this one is I'm supportive of it in the means that it relates back to the mate choice project and being able to select the fish. I'm in favor of some seed money into it anyway. We can't fund the whole thing, but I'd be in favor of funding it to some degree. I don't know what other discussion we might have, but we could look at these two projects we have then, the couch and banks, and what we want to do with them. If it's up for somebody wanting to make a motion of some way to handle it, then we can deal with it that way.
So I got a question. Sorry. I had a question for Michael Banks. So what would 50K do for you?
It would probably half fund a grad student, and I will, you know, I also had funds in the original proposal to pay some of the engineer's salary, so I wouldn't have as much for that, but 50K would be much better than zero. I mean, currently I'm bootlegging it on what I can and how persuasive. And you know, this engineer is very busy on other projects, and you know, if I get money to pay him, I have his ear, and I don't currently have a way of getting his ear. You know, I have a pin in his ear.
I think we need to find something for Michael.
Yeah, I have a mechanistic approach we can take. I think that might be able to get us there. We have 4 proposals that are still, you know, under consideration, including that from Michael Banks. What we could do is basically decide if we want, as the board, to request a resubmit from one or more of those 4. We can ask for that, and then with a maximum budget of 107K, that's what we have remaining in our bucket. And then we could evaluate those via email, and if we need to, for the public record, come together to do a quick vote to basically do the allocation. So for example, if there was an option where someone said they wanted to basically move to have us request Dr. Banks to resubmit a proposal in the range of 50K for us to consider, we could receive that offline, evaluate it, and then basically, you know, I'm not sure if there's a way to do voting without doing a public meeting like this, but otherwise, I think we're probably not. My understanding is it might be difficult for us to allocate against a partial proposal if we don't have deliverables that are set against that partial funding level. But I might be mistaken about that.
Thanks, Seth. But I think where we are is we've delayed these people a long time already that we're supposed to get back to them, and I don't think we should delay it any further. I think we better make a recommendation today and let the people move on.
I think the Couch proposal is pretty straightforward, which he could deliver with 50K.
If I could just step in, as I said, I believe earlier in answer to your earlier question, Seth, you kind of asked me this after the presentation. I think I can at least come forth with a prototype, which is what I'm desperate to have to use in November this year. I would do my best that way, regardless of the decision you make right now.
Sure. Well, the other option, I guess I just thought of would be, to your point, Scott, would be to allocate funding and ask the researchers to resubmit the deliverables based on that funding level, subsequent and pursuant prior to the release of those funds, instead of asking for a revised proposal. So I think that would be the other way to do it, which is we can allocate funds, but I think we have to tie that to a research plan that's laid out and that, is articulated, and the, fractional could be done that way instead of having to ask for it in offline consideration. So we could have that as a follow-up before the funding was released. I'd ask that to be included as part of any motion that was made.
That would have to be done to the contract part of it anyway through the university.
That's right.
Yeah.
Another thing we could do, those projects you might be interested in, Seth, they can always resurface in the next round of budgeting and research projects too.
Correct. No, and I think that's clear. The other ones are I think they need to be reformulated. I think we should encourage them to resubmit with some additional feedback, given the frame that we might want them to do on that.
Given the time we have and where we are now, I think I'll move to support Dr. Couch's project for 50,000 and Michael Banks' project for 50,000, and not fund the other two.
You need a second on that?
I need a second.
Second that. This is Tom Alcar.
I'd second that, absolutely. Michael? Yeah, one question, Michael.
Yes.
Are you going to use the Innovation Center to build all the parts that you needed that you've got going?
That was a current plan, yes.
Well, that's great. That's really a wonderful thing they've put together. I mean,
Yeah.
that really stands out.
Yeah. I'm very excited to use it.
So I would support that, and the part for I'd like to amend what will go in the motion to say that we'll basically request revised project goals from each that will then feed into the university contracting process.
Makes sense to me.
Yeah. Yes.
That's me. So we have a motion to fund Couch project 50,000, Michael Banks project 50,000, with the amendment to feed the other two projects back into the research protocol, I guess, for next go around, something like that.
Are we going to leave 7,000 in the budget? We have 107 to spend. Is that important or not?
Just leave the rest there, I'd say. Something might.
I think yeah.
More expensive.
The remainder is carryover.
It's worthwhile having carryover, but it also doesn't hurt, since there's over a year to go, to have a little wiggle room money in there in case one of the projects needs for something.
I'll just briefly add that total dollar amount is based on a projection as well. So if that budget
be a little conservative,
it would not hurt to be a little conservative here. If the revenue forecast falls a bit short, that $7,000 could be consumed by that.
Mark, could you could you officially repeat that motion, what you could write down there?
Sure. So my understanding is that we have a motion to support the proposal by Dr. Coach for $50,000, an additional $50,000 to support research by Dr. Banks. Was made and seconded. Have we closed discussion, or is there more discussion?
Still open for discussion.
Move to close discussion.
Do we need a
second that?
Yeah, I'd second that.
Okay. And a call for a vote?
Call for vote. Yes, go ahead. Call for
Move to
vote.
vote on the motion.
Okay. And I'll try and move quickly through this, not calling on folks who aren't present as best I can. Derek Wall.
Aye.
Seth Eisenberg.
Aye.
Tom Alcare.
Aye.
Ted Simon.
Aye.
Scott Starkey.
Aye.
Steve Jacobs.
Yes.
Pete Grundeck.
Aye.
Travis Mackey.
Aye.
The vote is unanimous. Mr. Chair, I'd like to make the declaration.
Oh. Motion. Ayes in favor of the motion.
Motion to adjourn.
Yeah, I just want to thank everybody for participation. I think we did pretty good clicking through this on time. There was a lot to cover, and I think it went well. I'm pleased with our results. So I thank you all very much, and make a motion to adjourn.
Second.
Yeah, second.
There's no further discussion. We'll say the motion carries and meeting is adjourned.